So thank you very much to Safine for organising this this meeting. It's been very interesting, and for also inviting me to tell you a little bit about what we do. So we work on a number of different ion channels, but today I thought I would tell you a little bit about our work on Kv7 channels. So I presume that many of you will probably know this, but the Kv7, or by the gene name, which is casein Q, are voltage-gated potassium channels, and each alpha subunit consists of 6 transmembrane segments, as shown here, with a pore region between S5 and S6. And the voltage sensor is in S4. And 4 of these subunits come together to form a channel, as shown here. So far, 5 subunits of the Kv7 family have been cloned, Kv7.1 to 7.5, and of these, 7.2, 7.3, and 7.5 are expressed in neurons either as homomers or heteromers, so 7.2 with 7.3 or 7.5 with 7.3. So when you express Kv7.2 and Kv7.3 together in cell lines such as HEK293 cells, and then apply depolarizing steps, you can record currents which are very slowly activating, and these increase with the amount of the step, as you can see here. And you can get an activation curve with a V-half of around minus 40 millivolts. Now, this resembled very closely to a current which had been recorded from SCG neurons by David Brown and Paul Adams in 1980, which was known as the M current because it was a potassium current that was inhibited by activation of muscarinic receptors. And so you can see that the biophysical characteristics of the Kv7.2, 7.3 expressed in the cell line are very similar to that of the M current. So these channels, although expressed in neurons, are also expressed in non-neuronal tissues, as shown here. Their 7.1 is expressed in cardiac muscle, skeletal muscle. You have 7.4, 0.2 and 5 and so forth. In the GI, vascular tissue, in the inner ear, there is a lot of Kv7.4. And mutations in these have been associated with many disorders. So mutations in Kv7.4 is to do with hearing loss, and there's a lot of very nice work by Thomas Jentzsch on this. And then also mutations in 7.1 have been associated with arrhythmias, LQT syndrome, and mutations in 7.2 and 7.3 have been associated with epilepsy. So we're very much interested in the function of these channels in hippocampal neurons. And so we previously showed, as has been shown here, that we have 7.2, 7.3, and 7.5 in these neurons. And so we wondered initially, what do they do here? So for this, we basically made brain slices and recorded from the soma of hippocampal CA1 pyramidal neurons. And under controlled conditions, this is a typical recording of a CA1 neuron, you can see that there is very little activity. This is done in the presence of glutamate, NMDA, AMPA kainate receptor blockers, as well as GABA receptor blockers. And then if you apply a series of hyperpolarizing and depolarizing current steps, you can record the voltage changes. So with hyperpolarization, you can see a sag here, which is due to IH, and with depolarization, you can initiate an action potential with depolarization. And this is the resting membrane potential, which is minus 65 millivolts. If you then apply Kv7 channel inhibitors, so in this case we've applied XC991, there is a depolarization of the resting membrane potentials, minus 58. And interestingly, we found that there was a little bit of spontaneous activity and also a significantly greater number of action potentials initiated with depolarizing steps. If you then hyperpolarize this back to the original resting membrane potential, which is minus 65, although you lose the spontaneous activity, you still have many more action potentials initiated with depolarizing steps. And the opposite happens if you add the activator, retigabine, which we got from neurosearch. So under control conditions, use minus 69 millivolts. If you add retigabine, it hyperpolarizes, and much more depolarization is required to initiate an action potential. If you bring the membrane potential back to its original, again, you still require a lot more depolarizing current to initiate action potentials. So how might inhibition of Kv7 channels be causing this spontaneous activity and increase in neuronal excitability? So obviously the depolarization in the resting membrane potential is likely to contribute to this. In addition, if you noticed that the voltage step, the voltage recorded with a current step is now much greater in the presence of XC991 compared to control, suggesting that the input resistance is much greater. That means that you need less voltage to initiate action potentials. But also, irrespective of that, the fact that you're having action potentials occurring at minus 58 millivolts made us wonder if there was something changed with the action potential shape. And when we looked at the action potential in detail, we found that the threshold of the action potential, single action potential, was much more negative in the presence of XC991 than in the absence. And in the case of retigabine, it was the opposite. So at the time when we were doing this type of work, there were a number of papers which came out which showed that Kv7.2, casein Q2, as well as Kv7.3, casein Q3, antibody staining was mostly in the axon initial segments and could be co-localized with a marker of the axon initial segment, which is anchorin G. So this is work from Ed Cooper's lab, Pan et al., but other people have also shown similar things. So, for example, here you can see that casein Q2, casein Q3, and anchorin G are all co-localized in the axon initial segments of CA1 pyramidal neurons, also cortical neurons, and CA3 pyramidal neurons as well. And in addition, what they showed, which was interesting, was that casein Q2 and casein Q3 in the C-terminus of the alpha subunits had a sequence which was very similar to that of the sodium channels, NAV 1.1, 1.2, and 1.6, which was important for binding to anchorin G. And they showed that if you mutated this, you could disrupt the trafficking of these subunits to the axon initial segments. So what we did was that because casein XC991 is bath applied, you cannot puff it very easily. It takes a long time to work. And because we were interested in understanding whether these Kv7 channels in axons were basically what was causing the spontaneous action potential firing, we designed a peptide which was a similar sequence to that which is shown here, the casein Q2. And we added it to the intracellular solution of the pipette. So in this case, we've patched onto the CA1 pyramidal neuron with the peptide, which we call anchorin G binding peptide, so ABP. And this is the initial recording. As soon as we go wholesale, you can see the resting membrane potential, minus 70 millivolts, and you require a considerable amount of depolarization to get action potentials. This is the trace, just with spontaneous. You can see there's very little action potential firing. With time, as the peptide dialyzes within the cell, you can see that the resting membrane potential changes, and now you start to see spontaneous action potential firing occurring. And then if on top of this you add XC991, you get further depolarization and further spontaneous action potential firing. And the data is actually summarized here. So this work suggested to us that there were channels in the axon, but also in the soma. And how many channels in the soma versus axon? So we basically collaborated with Michele Migliori to make a computational model of the CA1 pyramidal neuron. And we had recorded the Kv7 current from the soma and used the biophysical properties to insert the current both in axon and soma at different densities. And when we had 3 times as much current in the axon versus the soma, you can see the membrane potential is minus 65, and if we remove the current from the axon only, you get transient action potential firing. If you have 4 times as much current in the axon versus the soma, and you remove the current from the axon only, you end up getting spontaneous firing. And if you repeat this with only the soma, you don't actually get any firing at all. So this suggested to us that there was between 3 to 4 times greater amount of current in the axon versus the soma. So in the hippocampus, you have the pyramidal neurons, but you also have granule cells in the dentate gyrus, which are principal neurons. And the morphology of these neurons is quite distinct, as you can see here. This is a granule cell, and this is the dendrites, and it has a very long axon which connects to CA3 pyramidal neurons. This axon, unlike that of CA1 pyramidal neurons, is actually unmyelinated. And what is also interesting is that the properties of these neurons are such that they don't actually fire many action potentials at all. So these are in vivo recordings from Peter Jonas's lab from either anesthetized or awake animals, and you can see that there is very little spontaneous firing. The resting membrane potentials of these neurons is also very negative, around minus 80 millivolts. So we wondered, do these neurons have casein Q currents or Kv7 currents, and are they also in axons? So we made slices, and this is a slice one of our slices. So this is the dentate gyrus, which you can see here. And in collaboration with Ed Cooper, we did some staining from these slices, and you can see that there is co-labeling of the casein Q with a sodium pen NEV and also anchorin G, suggesting that these Kv7 channels, or Kv7.2 at least, is present in the axon initial segments of these granule cells. It was also in the distal portion of the axon initial segments. So how does the Kv7 currents contribute to the excitability of these neurons? So we made electrophysiological recordings from these neurons. So you can see the resting membrane potential is very negative. It's minus 81 millivolts or so. Again, we've applied a series of hyperpolarizing and depolarizing current pulses, and these are the voltage responses. And if you apply about 100 picoamps of current or so, you can get single action potential. So we added ABP to the patch pipettes again, and this time you can see that there is no change in the resting membrane potential. There was also no change in the input resistance, so the amount of the voltage change in response to depolarizing current pulse, but we had a greater number of action potentials. And if you looked at single action potentials, you can see that this is your control action potential, and 25 minutes following ABP dialysis, the action potential threshold is reduced considerably. If we add XC991, there is no further change, suggesting that in these neurons at least, there are no Kv7 currents in the soma, and majority of it is present in the axons, the mossy fibers. So in fact, early work by Ed Cooper when he was in Lily Jan's lab showed that these Kv7.2, 7.3 subunits were present throughout the mossy fibers, throughout these unmyelinated fibers. And one of the features of these is that you have axon terminals, giant axon terminals, onto CA3 pyramidal neurons, which you can actually patch. So we can visualize the giant mossy fiber terminals, which are synapsing onto the dendrites of these pyramidal CA3 pyramidal neurons. And so we basically learned from, we went to Peter Jonas's lab and we learned how to patch these terminals, and these are recordings from these terminals. And so what we do is we patch the terminals, we fill them with a dye, neurobiotin, and then we do post hoc staining, and this is one such mossy fiber bouton. So we made wholesale voltage clamp recordings from these initially, and what we did is that we held the bouton at a positive potential, minus 20 millivolts, to activate the Kv7 current, and then we applied a series of very long 2-second hyperpolarizing pulses in increments of 10 millivolts as shown here. So as you apply a negative step, the current deactivates and reaches, well, there is still amount of current left at minus 30 millivolts, minus 40. And if you then apply XC991, you inhibit this current, so you can see there is a change in the standing current. There is also very little deactivation now, and this is the subtracted current. So we could see that there is a significant amount of current present in these boutons. So one problem with doing wholesale voltage clamp is that you could be actually potentially measuring current from the axons segments which are sort of coming out of the bouton. So to understand whether this was the current was actually present in the boutons per se, we also did outside-out patch recordings. And so this is an example of an outside-out patch recording that we did. You can see there is still a considerable amount of current, about 75 picoamps or so. Applied the same protocol as above. You can get the deactivation, you can get inhibition of the current with XC991, and this is the subtracted current. If you look at the biophysical properties and the current densities as well as the kinetics of the current under either wholesale or outside-out patches, you can see that they're very similar. So the current densities are very similar. The inactivation time constants at minus 50 millivolts very similar. And also the activation curves are very similar. So we have a significant amount of current which is present within the bouton itself, which is what we're recording from. One thing that was very unusual was that the V half of the current, of this Kv7 current, was around minus 70 millivolts, which is a little bit more negative than what had been reported initially in SCG neurons, but is consistent with the biophysical properties of the current that we recorded at the soma of these granule cells and in hippocampal CA1 neurons. So what do the current do in these boutons? So again, we now made wholesale current clamp recordings from the boutons. So they have very negative membrane potentials, like at the soma, around minus 82 millivolts. And you can apply a series of hyperpolarizing, depolarizing steps, as you could see here. So the boutons have a very high input resistance of gigaohms, which has been characterized. And so you don't need like a significant amount of current to generate a single action potential. It's around, on average under control conditions, is around 80 picoamps to get a single action potential. When we added XC991, concentration of 3 micromolar, or linopodine, which is a second type of Kv7 inhibitor, and this works at 10 micromolar, you can see that there is actually no change in the resting membrane potential shown here. This is because these channels actually, these boutons have the inward rectifier potassium current at very high densities, and so that dominates, that dictates the resting membrane potentials of these boutons. But now in the presence of these Kv7 channel inhibitors, if you then put depolarizing steps, as shown here, you can see that you need less current to generate an action potential, so the probability of generating an action potential is enhanced. And also, with the depolarizing steps, the input resistance, so the amount of voltage recorded in response to a depolarizing step, is much greater. Okay, but in boutons, normally you don't have this type of, under physiological conditions at least, you don't have these steps. What you tend to get are action potentials sort of invading into the boutons. So we try to mimic this by giving very single, very sort of very short pulses, 0.1 millisecond pulses, to generate action potentials, single action potentials at different frequencies. So these are trains of 20 action potentials here at 5 Hertz. So under control conditions, you can see the black trace. This is you get action potentials with a very little after depolarization, or ADP, that follows this action potential. However, when you add XC991, the action potential characteristics change, so the action potential itself becomes much wider, and also there is an significant after depolarization happening. And you can see this also at 50 Hertz, if you do this at 50 Hertz or even 100 Hertz. In this case, what you're now seeing is that you're getting action potentials being generated on top of the after depolarization, so you sort of get a difference in the amount of in the voltage response here. So the action potentials are actually being generated at a much more depolarized potentials than under control conditions. Okay, so but there is a significant ADP, and also the action potential width changes as well. So why might this actually be happening in the bouton? So what we decided to do again was to construct a computational model, in this case a single compartment model, with the known biophysical properties of all the channels in the multi-fiber bouton that had been known, including, and we added our own Kv7 conductance in there. And we simulated an action potential. This is the black, which is a control, very small ADP. Now, when we have the calcium conductance and we remove the Kv7 conductance, you can see that the action potential width becomes greater, and there is a after depolarization that follows this. And if you look at the simulated calcium signals within the bouton, you can see that the calcium transient elicited with a single action potential is much greater when you remove the Kv7 conductance than in the absence of the Kv7 conductance. So accordingly, if we take out both the Kv7 conductance and the calcium conductance, in fact, you get there is no change in the action potential becomes narrower, and the ADP is reduced significantly. As we can see, it becomes much more negative. So this suggests that Kv7 currents here in these multi-fiber bouton terminals are restricting action potential induced influx, calcium influx. And that's normally due to N and PQ type calcium channels. So we just tested this by recording under control conditions. This is your normal control recording like we've shown before. And then we co-applied XC991 together with N and PQ calcium current inhibitors. And now you can see that the ADP is reduced significantly. There is no change in the action potential kinetics as well. So this suggests the Kv7 currents limiting calcium influx via these channels. So if it's doing this, it may be regulating synaptic transmission and action potential induced synaptic transmission as well. And to test this, what we did was we did dual recordings. So from the boot-on, we made cell-attached recordings to elicit an action potential, an action current, rather than an action potential, sorry. And we also did simultaneously patched onto CA3 pyramidal neuron under Wholesale voltage clamp. We had cesium in the patch pipette and QX314 as well. So now these are paired recordings, so you get an action current and you get an EPSC in the CA3 pyramidal neuron simultaneously. And if you then apply XC991, so this, because you've got cesium chloride here, you're blocking the postsynaptic Kv7 currents to begin with. So by both applying XC991, you should also only be inhibiting the presynaptic Kv7 channels, which are present on the boot-on. And you can see that you get an increase in the EPSC amplitude, and also the EPSC is now much broader than under control conditions. If you look at a train of action currents under control conditions, you can see that you get a paired pulse facilitation. So you get the second EPSC is much greater than the first EPSC in these recordings here. This is at 50 Hertz stimulation. And then with XC991, the paired pulse ratio is reduced considerably. So the first EPSC is much larger, and so the second EPSC is smaller. And so that's why the PPR is reduced. So this suggests that in the boot-ons, the Kv7 current is actually reducing synaptic release. So very quickly, it's like, no, okay. Okay, so very quickly, I just basically wanted to say that we also have basically, so let me just show you this. So this is basically the current which is recorded from the granule cell soma. As you mentioned, it's an M current, so it should be inhibited by OXO-M. And basically, in this case, it is inhibited by the muscarinic receptor agonist, which is OXO-M. And you can see it's inhibited, but now if you wash out with OXO-M, it basically is irreversible, which is very different from what you see in the soma of other neurons. So that means that although the somatic inhibition of somatic Kv7 currents is due to PIP2, we think that the inhibition of these axonal Kv7 currents by muscarinic receptor agonist doesn't involve PIP2, but involves an increase in calcium influx in the axon, and this is actually via T-type calcium channels, and so it's a different mechanism to what is present in the soma. So just very quickly, just to summarize, we think Kv7 channels predominantly localized to axons. In the axon initial segments, they regulate the action potential threshold. In synaptic terminals, regulate action potential induced calcium influx and synaptic transmission. What I didn't show you, but it's published work in 2015, endogenous acetylcholine actually activates muscarinic receptors to persistently inhibit Kv7 currents in axon initial segments by enhancing calcium influx via T channels, and that this might be a mechanism to physiologically regulate hippocampal neuronal activity. And finally, most of the granule cell work was done by Katushin. A lot of this work was all done with collaboration with David Brown, computational modeling with Mikkel, and antibody staining with Ed Cooper. I'd also like to thank Lee in Peter Jonas' lab, but now with Joseph Bischofberger in Switzerland, who helped us with the mossy fiber boot-on patching. Thank you.
Thank you very much. Questions for Marla? Yes.
Hi, Marla. That was a very nice talk. I just think, remember Jack Meller's talk yesterday? He was talking about cholinergic regulation, but I guess it was in C1 neurons.
Yeah.
this is cholinergic, but on CA3 through the granule cells.
No, in
In the
granule
granule
cells
cells.
alone.
So how does that all come together as a circuit then? Because it's all driven by cholinergic transmission.
So that is an interesting question. Yes. So we were very surprised, but we did manage to stimulate the afferents to get endogenous. And we see this here, but we also see a similar effect in CA1 pyramidal neurons, although I haven't shown this. But in CA1, I think it's a little bit different because you also get a lot of cholinergic afferents onto the dendrites as well. So you get a multiple response. In granule cells, you also have the muscarinic receptors on dendrites and soma as well. And in fact, you do get additional effects, but these effects are only amplified when we stimulate the cholinergic afferents in the presence of acetylcholinesterase inhibitors. So it's like prolonging the effects of acetylcholine itself. So we think that in the granule cells, it's really axonal is the majority of the effects. In CA1, also it's axonal plus maybe a little bit in somatodendritic as well. So in terms of the circuits, is probably promoting neuronal excitability, neurotransmission actually. What we don't know is how they regulate or what effect there is on the terminals, because there are receptors there on the terminals as well. So it's a bit complicated and it's not.
There was one last question just there. Jack.
Thank you. Yeah, really nice talk, Mala. What's your impression about what cholinergic activation does to synaptic transmission at the mossy fibre? Because a lot of people have found there isn't much effect, which you wouldn't expect to. I mean, that doesn't quite tally in with what you've found, right?
Yeah, so what I would predict is actually we did some EM staining for muscarinic M1 receptors, which I haven't shown, and they are present at a very low density on the CA1 on the mossy fiber terminals. But you also have metabotropic glutamate receptors, quite a large number on that, as you know. So I think that the Kv7 current can be regulated in two different ways, either via the mGluRs or the muscarinic receptors, similar to what you showed yesterday. So maybe with the muscarinic receptors, maybe you don't get such a large amount of change in the Kv7 current, but maybe it's via the mGluRs a lot. Yeah.
Thank you very much, Mala.
Sorry.
Very nice.